In the previous lecture 8, we started talking about experiments, how important they are. I've introduced the idea of what is a formal experiment, and we concluded the lecture with a sample experiment that was done many years back to prove that if we actually enhance how a piece of code looks like in an editor, the programmer will be able to understand the code better and will be able to comprehend the algorithm in front of him, and consequently it is actually a good idea that we have nowadays these IDEs like Eclipse and NetBeans and IntelliJ, because they really help us with the coding process. We'll continue on where we stopped, where what to do in terms of the data analysis. So at the end of lecture 8, I showed you the answers, the correct answers that was provided by each one of the participants to the questionnaire that was administered at the end of the experiment. And the question is what to do. And I went into an explanation that there are two types of statistics that we could perform. Descriptive statistics, which includes reporting mean, median, standard deviation, mode, and so on. And these will not be able to support any of our claims regarding the hypothesis we are proposing or regarding the null hypothesis. For that purpose, we have to refer back or use what's called inferential statistics. And I mentioned there are hundreds of tests that we could use as under the umbrella of inferential statistics, and the question is which test to use. Now, to select an appropriate statistical test, you need to consider three things. The first one is the type of the variables you're working with. We said we have independent variables, which are the variables you manipulate in an experiment. And you have the dependent variables, which are the variables you measure, what you measure from the participant. That's why it's called dependent. It's dependent on the performance of the participants. And you need to consider as well the research question. Is this x greater than y? Is this x related to y? Or is this x causing y? And you need to consider how you actually conducted the experiment, whether it is within subjects or between subjects. So let's see each one of those guys and what is the implication for the different choices we have in terms of the appropriate statistical test. The first thing we're going to look at is the type of the variable. We have two types of variables that we could be working with. What's called quantitative variables, and these are variables that represent numbers or values on a scale, like for example age or price or blood pressure and so on. Now, quantitative here has a specific meaning. It means that the numbers you have could be put on a scale, and between any two numbers there is a number that you might not have seen in your data, but that number has a representative member of the population having it, and it is just a matter of getting more participants to see any number that you would like or expect to see. So we're talking here about a continuous scale. Okay? The second type of variable is called qualitative or categorial, and in this case we do not have a continuous scale. Qualitative or categorial variables come in two types. Either ordinal, which means the values could be ordered. So, like for example, low income, medium income, high income. There is an order there. Or for example, level of satisfaction, from very interested to not interested at all. Okay? So if we can order the choices, then we will call it an ordinal, categorial, or qualitative variable. If we cannot order the choices, there's no meaning to ordering the choices, then we will call it a nominal qualitative variable. Things that we cannot order is, for example, gender, or team preference, or marital status. Okay, there's no order there. So if it's not ordered, then it's nominal. If it's ordered, it's ordinal. In both cases, this is not a continuous scale. What that means, there is no value defined between any two choices given to the user. For example, between income, low, high, and medium, there are no intermediate values. If you provide the user with an option to choose from low, medium, high, then you only have low, medium, high. There's nothing in between, even though conceptually we could think of something between low and medium. Okay? perhaps it's moderately low, but since we didn't ask the user about it, it does not exist because we're not even having a scale here. Same thing for Ahli or Zamalek, okay? There's nothing in between, okay? Same thing for gender or male or female or marital status. There is nothing between unmarried and married, okay? Obviously here you have to present the proper set of choices. For example, if you talk about marital status, you might want to include things like married, unmarried, divorced, widowed, and engaged, okay? But there's nothing between those labels. There's no scale in the first place to talk about that. The second issue you have to consider, or criteria for choosing a statistical test, is the type of the research question. Are we doing X greater than Y, like the example of the keyboard we looked at last time? Or are we doing an association, okay? Is X related to Y? Or are we saying is X causing Y, okay? Remember we said last time that you start by fixing the research question. So you cannot change it later on. And you will design everything based on the research question. And when it comes to pick the statistical test, you have to refer back to what you started with, because you're going to pick the appropriate statistical test based on the research question. The third criteria is how you conducted the experiment, how you collected the data. So you already mentioned that there are two ways, and I'm going to show you now a third way to collect the data. Technique number B, which is called paired. In the statistics world, in the experiment design world, it will be called within subjects design. In this case, all the participants will do all the experiment, or each one of the participants will do all the experiment. In the independent case, this is what we call in the experiment design between subjects design. Okay, so you distribute the trials between the subjects. So typically you will get a set of participants and split them into two groups. One group will do half the experiment and the other group will do the other half. And I went into a discussion last time about the example of pharmaceutical companies and why do they need to always do between subject design. There is a third variation, which is really a case of independent or between subject design, called matched design. And in this case, you're going to get a group of participants. You are going to split them into two groups. One group will do half the experiment, the other group will do the other half of the experiment. But when it comes to the statistical test, you are going to match subgroups within each group with a corresponding subgroup in the other half. Okay? A typical example here again, I could draw the simplest one to understand is from the pharmaceutical industry. When they develop a medication that is working at the gene level. So there are three main races on planet Earth of humans. Caucasians, which includes all of Europe, and North Africa all the way till Iran, and Africans, and then Asians. And by the way, Caucasians also include North America and Australia. Okay? So in terms of racial groups, these are the three biggest racial groups on Earth. If you go, for example, to Latin America, you will see mixes. You will see some people from an Asian background, like in Peru, or you will see, the Japanese in Peru, or you will see, Africans in Brazil and Argentina and so on, or you will see Caucasians, like in Argentina, where there's a lot of people from European descent. So, but the three major races are Caucasians, Africans, and Asians. When pharmaceutical companies are working on a medication or a treatment that will unlock certain genes or will affect how the gene behaves, or the gene itself either reject the medication or is slightly or affected differently by the medication, when they're doing the tests, they will get participants from the three racial groups or the three broad racial groups, and then when they're actually doing the analysis, they will match the Asians to the Asians, the Africans to the Africans from both sides, so they will get, say, 120 participants, say. 60 will do half the experiment, and the other 60 will do the other half. In the 60, there will be, in each side, there will be 20 Asians, 20 Africans, and 20 Caucasians. When they're doing the statistical analysis, they will match the 20 Caucasians from this side to the 20 Caucasians on the other side, and the 20 Asians on this side to the 20 Asians on this side, and the 20 Africans to the 20 Africans, and it goes like this. Okay, so this is called a matched experimental design. And it is again a variation of between subjects. So the takeaway is that to select a statistical test, you must have defined the research question, you know the types of your independent and dependent variables, and you know how the experiment was designed, and obviously this is all common sense. So let's look at what statistical test you're going to use if actually your research question is x greater than y. Okay, like the example of the keyboard you've seen earlier, or in lecture 8, or the example of the source code presentation towards the end of lecture 8. Okay, but I would like to say this is better than that. So this is how we talk about it. We say x greater than y. Now, the thing is with statistical tests, every statistical test has been designed to work with a specific research question, with a specific variable types, and with a specific experiment design. So, if you have one independent variable with two levels only, that's what it says here, two independent levels. So you have one variable, one independent variable with two levels, okay? And your dependent variable is quantitative. What you're measuring is quantitative. It goes on a scale, like for example, error rate or reaction time, okay? Or EEG signal. So if you have an experiment in which you have one independent variable and it has two levels, so we will call it two independent levels. That's the only thing you have in the experiment, okay? And what you're measuring is quantitative. And you conducted the experiment according to the between subjects design model, okay? Which means you got a number of participants and you split them into two groups, and every group will see half the number of trials, or half the experiment basically. If you have this, you will use what is called the t-test. Okay? What if you also had one independent variable, another situation, another experiment, you had one independent variable, and that independent variable had two levels. It has to be two, by the way, okay? And it had two levels. But you conducted the experiment according to the within subject model, which means all the participants did the whole experiment, which means every participant did the whole experiment. In this case, the statistical test to use is the paired t-test. Now I'm going to talk to you about what is this t-test, but I want you first to understand how we pick the test. You have to look at the research question, and then you have to understand how many, or figure out how many independent levels you have. If you have two, this means you only have one independent variable, because to have a variable you must have variation. That's why it's called a variable. And the lowest variation you could have, the smallest number of variation you could have is 2. So then we'll talk about two independent levels, okay? And you are measuring something that is quantitative, which means reaction time or number of errors. Then you do a t-test, either paired or t-test, the regular t-test, okay? Now what if you have something else? We will see what you're going to do. For example, if what you're measuring is ordinal or nominal, you're going to use another test. Okay? If you have more than two independent levels, which means if you have one independent variable with three levels, or you have two independent variables, and which means every one of them will have at least two variations, okay, two levels, which means you have in total 4 independent levels. So anything that's greater than 2, you're going to use something else. Now you might ask, why would I need ordinal or nominal? And actually, this is actually the data you will be dealing with if you design an experiment in which the data you collect, your dependent variable, is actually collected through a questionnaire, okay? Through a questionnaire, not by direct measurement, but through a questionnaire. So if you are into business and you are into user study or market study, you conduct questionnaires, and after you collect the data from the questionnaires, what you're going to do is you must have started with a hypothesis, and then you're going to take this data and put it into a software like SPSS, which you're going to use for your assignment, and you do a statistical test on it, okay? But you have to realize that the data from the questionnaire is either ordinal or nominal. When you ask about the gender, for example, this is nominal data. When you ask about, the income, you can order the income, low, medium, high, that's ordinal data, okay? So whatever that we talk about in terms of quantitative, because I'm more interested in doing experiments in HCI, and consequently, we collect numeric data, we collect quantitative data, but you could as well conduct a questionnaire and run the same or equivalent tests to t-tests to come up with a broad statement about what you're studying. So let's look at t-test. What's the story of t-test? Well, the first thing I'm going to tell you is T stands for Tukey. So Mr. Tukey is a famous statistician that he lived in the 1960s, and he came up with this very entertaining test. It's a statistical test, okay? I'm not sure how far you went in your statistical study or how much you remember or how much you would like to remember, but statistical tests are very interesting because they are used to make inferences about the data and not just to describe the data, okay? So I will show you quickly or briefly, what a t-test is, but I really don't expect you to go into the statistics for the purpose of studying HCI. I want you to know what is the input to every statistical test and what is the output, how you use the input, how you use the output, or how you feed in the input and how you use the output, and that's enough for me, bro, because the focus of this course is on HCI and is not on statistics, okay? But we still need to understand some basic stuff related to statistics. So in terms of input, think of t-test for now, it's like this white box that has some magical formula inside. It takes an input, three things. First of all, information somehow communicated to the t-test about the independent variable that you actually have manipulated. And you pass to it the data that you have measured, okay? The data that you have measured. So the dependent variable data, which is the measurement you made from the participants. And you pass to it something very interesting called the confidence level. The confidence level is the percentage below which you're going to reject the null hypothesis. If you don't know what I'm talking about, then you definitely need to watch lecture 8 first. So we said that we cannot prove any hypothesis. What we're going to do is we're going to work on the null hypothesis. The null hypothesis means the nullification of the hypothesis, the proposed hypothesis. So if I'm saying that keyboard X is better than keyboard Y, the negation is Y better than X. The not is Y better than X. But if I talk about the nullification, X and Y keyboards are the same. So the confidence level is going to be a percentage, and it is the level below which we reject the null hypothesis. So if we say the confidence level is 5%, what that means is if we get the probability of the null hypothesis below 5%, we're going to reject the null hypothesis and put forward our hypothesis. Now, why does the t-test takes the confidence level as input? Because every scientific discipline accepts a certain confidence level to be the standard. So for example, in HCI, we accept 5% to be our standard, which means if the probability of the null hypothesis is less than 5%, we're going to reject the null hypothesis. In pharmaceuticals, for example, because the implications is much more dangerous, people could die because of a wrong medication, they usually use the confidence level of 3% and sometimes even 1%. Okay? Now, this is actually fed somehow into the computations that the t-test makes, but I'm not going to go into that. Now, the output of the t-test is lots of stats, descriptive stats, because it's computing those stats along the way to be able to do what it's supposed to be doing here. But the most important output of the t-test is the significance, which is the probability of the null hypothesis. And this will always be reported as the p-value. P stands for probability. So, if you get out as an output from the t-test p equal to 0.03, that means 3%. That means the probability of the null hypothesis to be true is 3%. And since we have accepted the confidence level to be 5%, 3% is less than 5%, so we reject the null hypothesis. And we put forward our hypothesis. So the most important thing you look at in the t-test output is the sig value, okay, or the significance value, which will be reported as usually p or sig. Now, I'm not going to go into a lot of detail about how actually the t-test, and I don't require you for an exam or a quiz to actually know how this is computed, okay? But I'm just going to mention quickly what is this doing, okay? So it is simply, if you look at the numerator here, it's computing the difference between the means of the two inputs that is passed to it. Remember, the t-test is only applicable if you have an experiment with two independent levels. What that means is you're going to input two columns of data basically, and x1 and x2 are referring to those columns, and we are calculating the mean of x1 minus the mean of x2 in the numerator. Now in the numerator is actually the computation is a little bit more complex. We are calculating the standard deviation, okay, or the sigma squared, and then we take the square root of that and we multiply it by the square root of 2 divided by n, where n is the number of samples you actually had. Number of samples means how many readings you've actually made. Now, we are basically doing here, we are weighting the difference by the standard deviation between the two columns. Okay, if you don't get a sense of what this is trying to achieve, that's okay. I'm not going to go into the statistics of all of this, but I want you to understand that the computations will typically involve in differential statistics at least mean and standard deviation and some other variations as well. Okay, what we're trying to do here is something that I will clarify in the next slides, but with a more kind of visual example. So let's see our example here. Okay, assume I'm doing a within subject design experiment. Don't worry about the experiment, but let's say assume it's any experiment. Okay, so within subjects means every participant will do all the experiment. The blue dots here come from one level, exposing the participants to one level, and the red dots come from exposing the participants, the same participants, to another level. So let's put here some realistic, scenario. Let's say I am testing two keyboards, keyboard X and keyboard Y. So the experiment goes like this. Each participant will sit either using keyboard X or keyboard Y. I will put a piece of paper with him in front of him that contains some text, and I ask him to type the text that appears, either on the screen or on a piece of paper. It doesn't really matter, but they will type the text in front of them using the keyboard, and I measure how fast they finish. So I will give them a page of text, and I will repeat that obviously several times. We've already discussed this in lecture 8, but basically the test involves typing on the keyboard, and I measure the time it takes to complete a specific given paragraph, and I do that several times. Okay, and then I calculate the average for every participant. Okay, so this is our experiment. Two keyboards. The task is to type using each one of the keyboards. What I am measuring is the time it takes to finish the typing task. Okay, now I run the experiment, and then I plot the points. Say the red points is for keyboard X and the blue points is for keyboard Y. What you're seeing here, the dots, is actually the performance of the difference, the average performance or the average typing speed of each one of the participants. Okay? Now, I want you to look at this curve and tell me, is this one curve or two curves? If you actually keep looking, what you're going to do is you're going to try to fit a line on the top of each one of the sets. So you try to fit a line on the top of the red dots, and then you try to fit a line on the top of the blue dots. And you can easily see that you can fit one line on both of them. Okay? This is what I'm referring to. Okay? If I fit a line on the top of the blue dots, I get this curve. If I fit a line on the top of the red dots, I get that other curve. Okay? Now, I have put for you also labels for some of the participants, Ahmed and Huda. You see, Ahmed, when he was using interface A, his performance was where the arrow is pointing. And when Ahmed went and used interface B, which is keyboard X and keyboard Y, I'm referring to here, his performance didn't change a lot. In fact, it's the same level with some shift. So don't worry here about the x-axis and y-axis. I'm just showing you like a hypothetical scenario so you get an essence of what this statistical test is all about. Same thing for Huda. Okay, Huda performance didn't really change a lot. In fact, couldn't you argue that the performance of the two keyboards is the same, because I could fit a normal distribution on both of the blue and red dots. Okay, now the nice thing is here, of course, when I make up this example, I actually made it up so it looks like a normal distribution, and we've discussed normal distribution last lecture, and we said that it's quite important. So just by looking at the red dots alone or the blue dots alone, it's a normal distribution. Okay, now but really when you look at it and you keep looking at it, isn't there like one normal distribution that will fit, or one bell curve that will fit on the top of the blue dots and the red dots? Okay, isn't it really true? I mean, the overlap between the two curves is so huge that it looks like actually it is actually one curve. Okay, now statistically we would say that the population performance did not, or the population here, I mean the group of participants, the population performance did not shift because of changing the interface. Now, shift means what? Now, first of all, is there a shift? Yes, it's a little bit, okay? Ahmed was like a little bit to the left, now he's a little bit to the right. Huda was a little bit to the left, now it's a little bit to the right. But the shift is not drastic, okay? So we would say that the interface did not cause a major shift in the population performance. This is how we would describe this in the statistics world, okay? We're really looking at the same thing. Really what this is saying is that keyboard X and keyboard Y are the same, okay? It's the same normal distribution, okay? Now, let's look at this scenario, this second scenario. Again, let's assume that this is actually what we got, not the first curve at the top, but actually the second curve in the middle of the slide. Now you start to see something different. The blue dots is migrating away from the red dots. So statistically we would say there seems to be some kind of a major shift in the population performance due to interface B. Or you could say the other way around. There is a major shift in the population performance due to interface A. Okay? The points are getting further away from each other. It's not a complete migration. We're not really talking about two independent curves on the chart. But they're looking, a bit suspicious. I mean, you start wondering, are these two keyboards really the same, or what's going on? Now let's look at the third case. Now here is the third case. Okay? Now in the third case, you see two different curves. So I got the blue dots and I get the red dots, and if I try to fit a curve on the top of them, there will be two separate curves on the chart with minimal overlap. Okay? Now, this is different than the first one for sure. What was happening here, if the x-axis is time, obviously interface A or keyboard X is actually faster because all the population shifted. They are the same people, it's the same Ahmed, the same Huda. But when he was using keyboard X, the performance now is completely different from when using keyboard Y. Okay? Now, what I'm describing to you is the essence of the t-test. It is trying to figure out, are we looking at the same thing or we're looking at two different curves? And it will do that by weighting the difference between the means, or the mean of every one of the two data sets, weighted by the standard deviation. I'm here simplifying things a bit, but I want you to get the essence of it. Okay? In other words, we're asking, if you'd like, how much overlap is there between the performances, or between the curves, or how much there is no overlap. So when you get the significance or the p-value reported to be close to 1, like 0.9, what that means is the probability of the null hypothesis to be true is like 90%. What that means is there is no difference between the two interfaces. So if you get a number like 0.9, most likely you're looking at the first curve here as a result. Okay? But if you get it like 0.05, which is like 5%, or 0.04, which is like 4%, you're looking more towards the bottom curve, where the overlap between the two curves is so big that the probability of the null hypothesis, which is that the two are the same, is so low, because they're not the same, because there has been a shift on the curve, okay, or on the chart. There has been a shift, so the probability is actually quite low. And this is again the essence of the t-test. Okay, so we can summarize the t-test to be a test that compares the means of a continuous variable into samples in order to determine whether or not the difference between the two means exceed the difference that would be expected by chance. This is what the t-test is all about. Okay? In other words, what is the probability that the means are different, and they're not different just by chance. They are different because they belong to two to a big extent, independent normal distributions. As I mentioned, the t-test assumptions is that only two observations were made. I mean by that there are two independent levels, and that the observations are independent, okay, and drawn from a normally distributed population. So we've already discussed this condition of normality before in lecture 8, and we said that this is very important, that the data has to be tested for normality before running the t-test on the data. There is a number of types of t-tests. The first type we're going to look at is called the one-sample t-test. And in the one-sample t-test, this is a test if a sample mean for a variable differs significantly from the given population with a known mean. If you recall, when I discussed the research questions early on, I mentioned that that was also in lecture 8, by the way, in the beginning of lecture 8. I mentioned that we have 4 research questions, and the fourth one was, is the population performance that we are looking at is different from a known average. And if you'd like to test this, you can run a t-test to prove such a claim. I mentioned in lecture 8, if you recall, the example of going to a village and suspecting that everyone in this village is having diabetes, or at least everyone you meet is having diabetes, and you wonder if the average number of diabetic cases in this village is the same as the national average, say in Egypt. So what you run then is you collect data about the participants, maybe perhaps their insulin level, and you compare that to the Egyptian average insulin level. And then you can claim either that they are different or they are similar to the rest of Egypt. So this is what's called a one-sample t-test. Now in the independent or unpaired t-test, okay, what we're talking about here is that we are doing an experiment between subjects. So we are distributing the conditions on the participants, where every half of the participants will take half the experiment or will do half the experiment. Okay, that's why it says at the bottom 2 scores are taken but from different groups. If you have that, the test actually to run, it's a t-test, but it's called an independent or unpaired t-test. This it's the same as what shows up in the table that I was showing you earlier. Okay, so for example here, if you look at the example at the bottom, it says study effects of age on swearing. Okay, so the assumption here is that there is a relation between how old or how young someone is and the language they use. So you get 20 10-year-olds and 20 15-year-olds. Okay, now you will record the swearing hour in the swearing amount in 2-hour period, and what you're really comparing here is the effect of age. So this has to be done between subjects. You're going to get one group that's 15 years old and another group that is 10 years old. Okay, you cannot do it within subjects here. You have to split them into 2 groups, one 15 and the other one is 10 years old. Okay, now because this is between subjects, what you're going to do is you're going to do what's called an unpaired or independent t-test. Now how do you differentiate between these tests when you actually run them? In any statistical software like SPSS or SAS, you will get options to choose the type of the t-test you're going to run. So you'll have to just check, that this is actually the independent or unpaired t-test. Now the third type is the paired t-test, and this is, as you probably guessed, is when you use the within subjects design paradigm, where everyone in the participants group you're going to recruit will do the whole experiment. Okay, and this is what generally is called just the t-test. So a test if the population means estimated by dependent samples differ significantly. Okay, so here is an example. 20 people tested on attitude towards recycling after watching a nature's video. Now the independent variable is the video. That's what you are actually manipulating. The dependent variable is the score on the test. So what you're going to do here, you are actually going to give them the test before and after the treatment. So first you give them the test, and then they watch the video, and then you give them the test. Okay, and see if there is a change in the attitude just because of watching the video. Now here it's the same group doing the test before and after the video. So this is going to be a paired t-test. Here is a question from a previous exam about experiment design. So it says a company invented 2 new mouse designs as shown below. Your task is to design an experiment to find which one is more efficient to use. The questions were: describe a trial, what's your hypothesis, what's your null hypothesis, what are the independent and dependent variables, and which statistical test you would use. Okay, so let's think about this. So I have here 2 mice, I'm going to compare them, and somehow I have to compare them for efficiency. So if you think of efficiency in terms of a mice, we've already studied motor laws, we've already studied Fitts law, so I know that if this mouse is actually faster, one of them is faster than the other, then for any target acquisition task, it's going to be faster. So I need to set up the trial so that I do what's called a target acquisition task. So the cursor perhaps will appear in the center of the screen, and then a target will appear to the user, and I will tell him about the target. Maybe it's like a red circle, and I tell him, when you actually see the red circle, go and drag the mouse all the way to the red circle and click on it. And I measure the time it takes to actually, starting from the center, he will go to the red circle and click on it. Okay, so this will be the trial in my experiment, just to measure efficiency. If indeed one of them is faster than the other, you will be able to do the acquiring of the target faster. Now, what's your hypothesis? I will assume here anything. Perhaps I will assume that the mouse on the right is faster than the mouse on the left. What's the null hypothesis? That they are the same. What's the independent variable? It's the mouse type. And what's the dependent variable? It's the time it takes to travel to a target. Now the question is, the last one, which statistical test would you use? And since here we have only 2 variations, mouse 1 and mouse 2, so this is going to be t-test. And if I have specified somewhere along my explanation that I'm going to be doing it within subjects, so it's going to be paired samples t-test, or just paired t-test. Now, here is some good news. It's not only that t-tests are used in all scientific disciplines whenever you have 2 levels, or 2 independent levels, or 2 dependent levels as well. It will work, as we mentioned. It's not just that. When people use the statistical tests, there is a template in which that they use to actually report what is the output from the statistical test. So in order to introduce some kind of standardization among all scientific researchers, whether it's physics, chemistry, biology, pharmaceuticals, food science, and so on, computer science, HCI, we all use the same terminology, we all use the same writing style. And that's why I was telling you in lecture 8, if you learn this stuff, you can go and read any paper later on and figure out what they're talking about. You might not know the extreme details or the fine details that actually will require you to be an expert in the field to understand, but when you look at the experiment they conducted, you can immediately recognize what was going on. So for any research paper, when you actually report the results of a t-test, now how you're going to run the t-test, you will see that in the tutorial. It's going to be a software that you plug in the numbers and you say run t-test and you're done. Okay? Now the question is how you report the results, and again there is a standard way. So this is what you're looking at here is the standard way. You will say there was a significant difference. That's if there's actually significant change, or in other words, significant mean here the probability of the null hypothesis is less than 5%. So there was a significant difference in the scores for mouse 1 and mouse 2. And you report the mean and the standard deviation between brackets. And then you say t, you're referring to the t-test. You put in the number we're going to talk about, and then you put in equals, and then you grab the t-value, okay, and then comma, and then you say p equal or p less than, and you specify the probability of the null hypothesis. So obviously here we send significance because the probability of the null hypothesis is 0.02, which means it's 2%. So since it's less than 5%, we're happy, we reject the null hypothesis, we're assuming significance, and we will put forward our hypothesis. Now, what remains to explain here is what is this 8 and 2.89. Now, 8 is the degrees of freedom. Okay, I don't really require you to understand exactly how the degrees of freedom are computed, but I want you to understand the general idea of it. The general idea of it is that the number we're looking at, regardless of what could change in the values that we obtained from the users, will stay the same. Okay, so the degrees of freedom are generally n minus 1, where n is the sample size. Okay, it's used in determining the t-value, which appears to the right, 2.89. Now, you got 9 participants. You measured them. Now, and you got some results. The question is, if I take away from the 9 participants 3 and bring new 3 participants, so the data will change. If I take 4 and I bring 4 new participants, the data will change. But the question is this changing to the point that I will see completely different results? And the answer is no. If your sample size is correct, regardless of how many participants you're going to replace later on, how many participants data you're going to replace later on with data from new participants, you still get the same results. Okay? Obviously with some variation, because the new participants will perform slightly different than the old participants, but you still get significance. Now the 2.89 is a long story, so I usually don't go into it in the HCI course, but I would refer back to you the tables that you have used in the probability and statistics course that are known as the stats tables. We are getting that value from there. Okay, so if you actually don't recall what that is, I'm sure every time I taught this course and I asked students, do you remember these tables and how they work? And students asked yes, would answer yes. These tables are tables full of numbers, and you pick up a number by checking the x and y axis. I hope now you remember what I'm referring to. So this is where we're getting these numbers from. And this is just an optional slide if you're interested in understanding degrees of freedom, but I don't usually ask you about it in the midterm or an exam, because it's beyond the scope of the course in terms of the statistical level that you need to be aware of to actually answer any questions about this accurately. But I want you to get the basic idea, because we keep including the degrees of freedom in our result reporting. Okay, so it represents how many values involved in the calculation have the freedom to vary. This is the very broad definition. Okay, degrees of freedom equal to number of observations, okay, minus the number of parameters or levels. So in the t-test you have 2 parameters, okay, and consequently you will have n minus 1 as the degrees of freedom. Okay, now let's look at the example at the bottom. For n equal to 4, so your sample size is 4. Okay, now, and these are the values you got. The degrees of freedom is going to be n minus 1, which is 3. What does that mean? It means we can change any 3 numbers from the numbers you're seeing here and still get the mean equal to 20. Okay, but if we change the 4th, we can't. I'm just trying to communicate to you the essence of what does it mean to say degrees of freedom. Okay, so here I have sample size of 4 participants, and these are the readings I got from them: 15, 30, 25, 10. Okay, the mean of these guys is 20. What degrees of freedom is telling me, if I remove 3 of these participants and get another 3, okay, would I still get the 20 as the mean? And the answer is yes. Okay, so what this means that the answer I got is a global answer. It's not particular to the group of participants I actually run the experiment on. Now let's move on. We already talked about the situation where we have 2 levels. What if we have more than 2 levels? What we're going to do is we're going to run another test that acts very similar to the t-test with a slight difference, and this test is called ANOVA, or analysis of variance. So let's look at the ANOVA. So what is the ANOVA? The ANOVA is just like the t-test. It takes some inputs, which includes the independent variable data, okay, and the confidence level, just like before. But now we don't just have 2 levels, we have many levels. We have 3 or more. So if we talk about having 3 levels, this means we have 1 independent variable with 3 levels. If I talk about 4, it could be like 1 independent variable with 4 levels, or 2 independent variables, each with 2 levels. Because the minimum I could have for an independent variable is 2 levels. Now, what is output from the ANOVA is similar to the t-test. We're looking for the significance, or the p-value. This is the probability of the null hypothesis. Okay? But things a little bit here complicated. Let's see why. If you have just 3 levels, like one independent variable with 3 levels, if you go back to the example I introduced experiments with, which is the keyboard example, the Chinese keyboard, Microsoft keyboard, and X keyboard, let's call them here A, B, and C. If I run ANOVA and it gives me significance, what that means is there is something significant, but it doesn't tell me which pair are significant or whether they're all significant. So if it gives me significance, it might be that A and B are the same, which is the figure on the right, with the figure on the left, okay? But C is different from A and B. So I could treat A and B as the same. So in our example, if we're going back to the keyboard, it could be like the Microsoft keyboard and the good old Chinese keyboard are the same in terms of the performance. But the X keyboard, which was the most expensive one, is actually different. So what that means is Microsoft is making a scam by selling that keyboard for £500 while the Chinese replica is for £50. Okay? Now, it could be also that the 3 keyboards are different. The Chinese replica is really a bad keyboard, and you will be slow typing when you type on it. Microsoft keyboard is slightly better than the Chinese keyboard, and the best keyboard is actually the X keyboard. It's the fastest keyboard to use. So in this case, the 3 are statistically significant from each other. Now the problem with ANOVA, it's not really a problem, it's a side effect of how actually it works. It will not tell you what's going on. It will tell you there's something significant, but it will just stop there. So what that implies is you have to figure out which ones are significant. Which case are we talking about? Are 2 of them are similar and they are different from the third, or the 3 are different from each other? And if 2 of them are similar and they're different from the third, which 2 are actually similar? Is it A or B, or A or C, or B or C? Okay? So let's recap. ANOVA, analysis of variance, used when you have more than 2 conditions in an experiment. Okay? And there are really 3 types. One way ANOVA, repeated measures ANOVA, and factorial ANOVA. So let's see what these guys are. The most famous type is actually called repeated measures ANOVA. Now you have one independent variable, 3 or more conditions. Okay? You repeatedly measure from the same participant over and over again. This is used when you have within subjects design in an experiment. Okay? So because I'm saying you will measure from the same participant over and over again. So this is within subjects design. Now, again, it will tell you, whether the null hypothesis has been invalidated or not, but it will not tell you where is the invalidation. Okay? So what is required is, if you actually get significance from ANOVA, what you do afterwards is you go and run what's called post hoc tests. In post hoc tests, post hoc means after the fact. So in post hoc tests, what you do is you compare every pair together. Okay? It is somewhat similar to the idea of a t-test, except that it is not a t-test. It's a modified version of a t-test, if you like. Okay? But we cannot call it being just a t-test. Okay? There are many actually variations of post hoc tests depending on the type of data and the type of research question and the nature of your data. So I won't really go there because that's like a big topic that will require a lecture by itself. But I want to tell you that there are tests that are done after the ANOVA produce significance, and those tests will tell you which pair are significant and which pair are similar out of all the pairs you have. Okay? Now, so post hoc tests in the basic implementation or in the basic idea, they are basically doing pairwise comparisons. They would compare A and B, A and C, B and C. And if you have 4 levels, let's say you have 2 independent variables, independent variable 1 has A and B, independent variable 2 has B and C, they will do all the combinations. A and B, A and C, A and D, B and C, B and D, and C and D. Okay? And they will tell you which ones are statistically similar and which ones are statistically different. So there are many types. Okay? I've just listed here some of the names that you need to be aware that they exist, but again, I will not go and describe when to use which one, depending on your project or your assignment, depending on what I ask from you. You might be required to actually go and read about one of those to understand should you read it, should you use it or not. Okay? So the most famous one is Stuckey, followed by Bonferroni. Pascal Wallace was also very famous. Okay? And you will see these names in research papers, whether you're reading a research paper from the business community, or you're reading a research paper from food science community, you will see them. Like you will see them saying, you know, there was a significant difference between variable x and variable y. T-test produced significance. Here's the degrees of freedom. Here is the t-value. Here is p less than 0.05 or 0.03, and that's if they actually doing a t-test. And if they are doing an ANOVA, they will tell you, you know, we conducted repeated measures ANOVA, and the independent values were so and so, and independent values were so and so, and the repeated measures ANOVA was followed by the Ponferroni post-hoc test. Okay, so these names are very, very popular when you do experiments. Okay, now, and they all, by the way, are very similar to t-test, but not exactly. You know, there's always this slight twist on them. But again, I won't go into detail here about this. So here is an example of what I was referring to. Okay, if you consider a 3-level independent variable where you find that p is significant. Okay, you need to assess all possible pairwise comparisons: 1 versus 2, 1 versus 3, 2 versus 3. That's what you get from a post hoc test. Okay, it will tell you if there is significance, since you've already found significance, which one is actually significant and which one is not. Now let's move on to one-way ANOVA. So we've discussed repeated measures ANOVA. What about one-way ANOVA? Okay, one-way ANOVA is the equivalent to repeated measures ANOVA, but it is actually for between subjects design. So this is actually the case where you split the experiment the participant into 2 groups, and you divide the experiment between the 2 groups. So every participant will see half the experiment. So if you do that, then you are actually going for one-way ANOVA. Okay, and just like I mentioned with t-tests, if you're using a statistics software to actually do these tests, then it's almost like using Excel. You put in the data, and then you click from a menu saying run this test, and that's basically it, and it gets, the magic works. Now we come to the important one as well, which is factorial ANOVA. So this is the third type of ANOVA we're going to see. Now factorial ANOVA is very interesting because it's kind of a complex statistical test in some aspects. Okay, let's say you have 2 or more independent variables in an experiment. Like for example, I have here gender and age. I would like to test the effect of gender and age on humor. Okay, basically I'm asking the research question, is males above 30 more funnier than females above 30, something like this. So I bring participants. They are of different genders, male and female, and they are of different ages, 10, 20, or 30 years old. Okay, and I show them a comedy show, like Bill Cosby or any comedy show. Okay, and obviously I have to fix the comedy show because they will all be supposed to be exposed to the same thing. Now what I do is I measure how many times they laugh during the show. Okay, now let's think about statistics. What can we do? We have 2 independent variables, gender and age, and we have a dependent variable that is 1, which is the number of times they laughed during the show. So how do we do the analysis? Well, you could actually split the whole experiment data into 2 portions. One portion you have the gender, male and female, and the frequency of laughing, and you deal with that separately. Okay, and then another portion you have age, or another set of the data, you have the age and the frequency of laughing, and you treat that as a separate experiment as well. Okay, so that's possible, but there is a problem. When we start to have more than one independent variable, usually what happens is there will be what we call interaction between the independent variables levels. What that means is you could find that males who are 30 are funnier than females who are 20, or females who are 30. So it's not enough to be 30. You have to be male to be more funnier. Okay, to be different statistically. These are called interaction effects. That there are interaction between the levels of the independent variable, where if you pick one level from an independent variable and you pick another level from the independent variable and you compare it against the other levels, you see statistical difference. But if you remove one of the levels and you just work with the other one, you will not see statistical difference. Okay, now so theoretically we could run this analysis as 2 separate experiments: gender and frequency of laughing, and age and frequency of laughing. But we are denying ourselves to discover interesting relations or interesting interactions between gender and age. That if someone is old and he's a female, he's funnier, okay? But if he's old and he's a male, he's not funnier than, say, a younger age, okay? So this is what factorial ANOVA is for, when you're interested in not just in the main effects, which is gender and age, but also in the interaction between the main effects levels. Okay, so this is what I try to say here, that why do you want to look at two at the same time, okay, instead of conducting two separate studies. Okay, you want, when you're doing the analysis, to finish the analysis. Now you're going to do that twice the effort. Okay, so being more efficient, you definitely want to do factorial ANOVA. Okay, also being more interesting in the results in front of you, or being more curious to actually uncover hidden relations between the levels of the independent variables would take you towards factorial ANOVA, because factorial ANOVA will reveal things you cannot find without it. Okay, and it is common sense, actually, as I mentioned earlier, that once you start to have more than one independent variable, to expect some interaction, to expect things to actually be mixed up together. Like you get level, this specific level from this independent variable, another level from another independent variable, a third level from another independent variable, and now you have significance. Okay, like for example, if you decrease the light in a room, okay, the light intensity in a room, and you make it colder, okay, and you decrease the humidity, then participants are more likely to fail to sleep, to fall to sleep. Okay, they will sleep. Okay, but you need the three. You need to decrease the light, not increase the light, decrease the light. You want to increase the temperature, and you want to decrease humidity. Okay, so people will sleep faster if the three conditions are met. How would you actually figure that out? Through the interaction between the independent variables. Okay, now in the factorial ANOVA world, each independent variable is called a factor. Okay, so this sometimes will be called multi-factor. Okay, and each factor, obviously, or independent variable, has to have two levels at least. Okay, now the thing is, when you run a factorial ANOVA experiment, you have to have a complete design. What do I mean by complete design? It means each level of one of the independent variables must occur with each level of the other independent variables. So usually when you run factorial ANOVA experiments, you have a big number of combinations you have to consider. Okay, so if you have three independent variables, the first one has two levels, the second one has three levels, and the fourth one has four levels. What you're talking about is 2 by 3 by 4 combinations. Okay, 2 the first, 3 the second, 4 the third. 2 by 3 is 6, 6 by 4 is 24. You've got 24 combinations that you want to have, that you need to run the experiment with them, or in other words, your participants must do all the 24 combinations. Okay, so if the first one is light intensity, you have high and low. The second one is temperature, you have high, medium, and low. And the third one is humidity. You have 4 levels of humidity: 25%, 50%, 75%, and 100%. You want all the combinations. This is what we make. But this is what we mean when we talk about a complete design. So you gotta cover all combinations, including low light intensity, low temperature, and 25% humidity, and low light intensity, low temperature, and 50% humidity, and it goes like this for all of them. Okay, this would be a complete design, and you need that for factorial ANOVA to work. Okay, now here is another experiment, again just to get you thinking in this. Examine the effect of sleep and caffeine on the reaction time. So I will get a group who slept 2 hours or slept 8 hours. So I will get participants who either slept 2 hours or they slept 8 hours, and I will get them drinking zero caffeine or 3 Cokes, which is a lot of caffeine. And to measure the reaction time, what I'm going to do is I'm going to make them play a game, and I'm going to measure the reaction to the events in the game. As long as they go through the same path in the game and they see the same characters and they're required to do the same actions, this is going to be a fair platform to assess the reaction time. Now, the factorial design, okay, I will randomly select a participant or subject for each condition. Okay, The nice thing about factorial design, it will not just test if the two independent variables, which is caffeine and the number of hours of sleep, are significant or not, or they have an impact on the reaction time in the game. It will also test the interactions, which is the interaction of sleep and caffeine. So let's see how that is done. Okay, now first, before we see that, factorial ANOVA will give us what's called main effect analysis, which is the effect of the independent variables, okay, of the levels of the independent variables. Okay, now when it's doing that, it's treating the other independent variable as a transparent entity, as if it doesn't exist. So it will collapse the data across the independent variable that we're not interested in and keep the data for the independent variable we're interested in, and then we'll perform the analysis. Okay, this is what I'm referring to. If we analyze the effect of Coke alone, we don't worry about sleep. So the comparison is going to be 0 Cokes versus 3 Cokes, regardless of the number of hours they slept. And if we analyze the effect of the main effect, sleeping hours, we're going to compare 2 hours to 8 hours, and we are completely going to ignore, the amount of Coke they had. Okay, this is the idea of what's called collapsing a dimension to work with the other dimension. And this is what I was describing earlier, where if we study the effect of the amount of Coke they drank, we're going to ignore the number of hours they slept. Okay, we're just going to collapse the dimension of the hours. And in the previous slide, I was collapsing the dimension of the Coke. Okay, now what's interesting is that we're going to also study this case, the combinations against each other. 0 Coke in 2 hours, 0 Coke in 8 hours, 3 Cokes in 2 hours, and 3 Cokes in 8 hours. And if there is a significance in one of those combinations compared to the other 3, it will be reported by the factorial ANOVA. Okay, and this is called the interaction effect analysis. Okay, so this is what factorial ANOVA is all about. Now, how do you report factorial ANOVA? I have put here for you a link. You should check it out from the lecture slides about the reporting of factorial ANOVA. It will look very similar to the t-test, by the way, so it's not really something that should be surprising to you, except that some of the terminology changes. What about the rest? We were talking about the dependent variable being quantitative, and you having 2 levels or more. So 2 levels we said t-test, more than 2 levels we said ANOVA. What if you're having ordinal data or nominal data? There is an equivalent statistical test to each one of the ones we have described, but for the different dependent variable type. So if you're working with ordinal data that is ordered, and you're doing more than 2 groups, you are going to go for, or 2 independent levels, you're going to go for Kruskal-Wallis. Okay? If you have paired data, you're going to be Wilkinson. And it goes like this. So in any statistical software like SPSS or DAS, you just pick the right test. You just go for run Wilkinson after formatting the data in the way Wilkinson test expects. And then you get the output as saying the significance is so and so. Okay? If you get interested in these tests, obviously I invite you to check any statistics books about the differences between them, and you will find lots of resources online about that. But again, this is not part of an SCI course because it's kind of outside the scope. We're using these tests as black boxes. We pass them the data and we get the significance out, and that's what we care about. So here's the takeaway. For data analysis of an experiment where the research question is x greater than y, all applicable statistical tests are the same in what they do. Okay? So whenever you do x greater than y, they're doing the same thing. They're trying to figure out, are we talking about population performance that overlaps, or there is a statistical significance, or in other words, there are 2 different curves that we are actually considering. Okay, that's what I try to say here. They tell us if there is a statistical difference between 2x and y or more levels or not. It is the significance or sig that we look for. That's the probability of the null hypothesis being true. If less than 0.05, we reject the null hypothesis and put forward our hypothesis as an alternative explanation of the recorded data. So this is a summary of everything I have mentioned earlier. So what about x causing y? So I'm going to go faster now. Now you got the basic idea. Okay, so here is, if you're trying x causing y, things like smoking causes cancer, or using the computer causes neck problems, okay, neck injury. So how do you actually measure that? You have to ask yourself, what is the dependent variable, okay, and what is the independent variable type, and based on that you pick the test. So if you think of the traditional case in SCI, if I have the dependent variable quantitative, what I'm measuring is quantitative, like price, for example, or blood temperature and so on. Sorry, body temperature, not blood temperature. Okay, you don't have to actually take someone's blood out to measure his temperature. Now, and so you're measuring quantitative as well, then you do something called linear regression. If you're doing quantitative and categorical, you do ANOVA, the one we described earlier. If you're doing categorical and categorical, where you have something called multiple logistical regression, that could work for you. Okay, so let's understand regression as an idea, because we actually could use it in several places, as we will see later. So what is regression? Regression is another statistical test. It's kind of interesting because what this actually test is telling you is something different from ANOVA and t-test. What this will tell you is how much the input data is related together. How much are they in sync together, if you like? How much they are similar, together to some extent. What you get out that is important from the regression is the R value. Okay. Now the R value will vary from -1 to 1, and we will see what that means. And then you get lots of other stats. But really, whenever we run a regression, we're looking for the R. You go and check out the output of the statistical software you're using, whether SPSS or DAS or whatever, or R, and you just look for the R. By the way, R is it's a very badly chosen name. It's a name of a very popular nowadays statistical and machine learning package. It's called R. It has nothing to do with the R value of regression. Okay. So, what is regression? A method to estimate the magnitude of the effect of one or more independent control variables on a dependent variable. Okay. Remember, we're saying here x causing y. And when we go to x related to y, we will see the same thing, by the way. We will see the same regression appearing again. So it's very common to use it, okay? So what is regression mathematically? It's a method that indicates a mathematical relationship between a dependent and one or more independent variable, okay? If that relationship cannot be found, the R is going to be 0, okay? There is no relation between what has been measured and what has been manipulated. But if there is a relation, the R will not be 0, it will be something else. It will vary from -1 to 1. So this is what I was telling you about. R value is very important. If R is equal to 0, that means the regression was not able to find a mathematical relation between the data you measured and the independent variable you have manipulated. If R is equal to 1, that means the dependent variables, or the dependent variable that you measured and the independent variable that you have manipulated, are actually very, very related. In fact, they have such a strong relation between them that if you make any change in the magnitude of one of the independent variables, you will see a corresponding change with the same magnitude in the dependent variable. Okay? So let's say you're trying to investigate if, increases in the temperature makes people more aggressive. Okay? If the R is 1, that means with every degree of increase in the temperature, there will be a degree of increase in the people's aggressiveness. So you must here assume, I'm assuming that there is a scale for aggressiveness, okay? And you will be able to map the impact of 1 degree change in the temperature to a unit, some unit or some units to on the scale of aggression. So there's a one-to-one mapping between the change here, or the magnitude of the change here, and the magnitude of the change over there. The magnitude in the change of the independent variable and the magnitude of change in the dependent variable. Okay? It's one-to-one basically. So obviously that's too ideal. We rarely see this where R is equal to 1. But what that tells you also is that between 0 and 1, the more the value, the more these two things we're studying agree together in terms of the magnitude of change. If this changed this much, that amount, that exact amount, or close to it, will change in the dependent variable. If we change in the independent, it changes in the dependent. Okay, what we're recording from the dependent. So this is what the R, between 0 and 1 really means. Okay. Now, if R equal to -1, it means they are actually strongly related, but they are completely inverse. If you increase one, the other one decrease, and with an equivalent amount. Okay. When R is equal to 1, not -1, when R equal to 1, if you increase one, the other one will increase with the same magnitude. But if R is equal to -1, when one increase, the other one decrease, and it will increase with the same magnitude of the increase. So if you increase here with 2 units, the other one decreases with 2 units. If you increase here with 5 units, the other one decreases with 5 units. Okay? So they agree on the magnitude of the change. Now, what if R is between 0 and -1? It means the relation is inverse. When this increase, the other one decrease. but it's not going to be with the same magnitude. Okay, so maybe 1 degree of increase in temperature will result in a jump of will result in a reduction in the aggressiveness scale by 5 degrees, for example. So there is an inverse relation, but it's not the same magnitude. Here is 1, the other one is 5. Okay, so this is what regression is all about. Okay, uncovering relations between data sets and really uncovering the changes that happen in a data set and the impact of those changes on the other data sets. Now, by the way, we have so many types of regressions. Okay, as far as I remember, there is perhaps 7. Okay, but I'm not going to cover them because there is no time in this course to actually cover all the regression types. But I want to tell you that there are actually a number of regressions, and each one is used in a certain context. And you will see this later on when we talk about X related to Y. You will see other types of regression. Okay, the most famous type is called linear regression, which is the one that almost most people use because that's the one that kind of first of all it's easier to understand, and second that's what people are interested in. They would like to figure out, if you have quantitative data, of course, and you're manipulating quantitative independent variable, you would like to know when you increase the the independent variable, what's the impact on the dependent variable. So numerically, because they are quantitative, we can use a linear regression. What about X related to Y? I was just describing that. Well, again, you're going to check the type of the dependent and independent variable, and you're going to pick the appropriate statistical test. So you see here, logistic regression, for example, is appearing, which is another type of regression that's doing the same thing that we're describing with a slight twist. Okay, and you will use that if your dependent variable, the data you're collecting, is categorical. Okay, regardless whether it's nominal or ordinal. Okay, and if you're manipulating, what you're manipulating is quantitative as well. Okay, so you use logistic regression, and the rest are kind of spelled out here. So the takeaway is that for data analysis of one experiment where the research question is X related to Y, all applicable statistical tests are the same in what they do. They tell us how much is X related to Y through a computed index, example the R, that varies from strong correlation to not at all, and from positively to negatively correlated. Okay, so this is a summary of everything I described earlier.